welcome to part 2.
Thank you. Nice to be here.
Good. The floor's all yours.
Thank you very much. After we had the first interview, several of my colleagues and some other people had specific questions, and that's why I thought we can do the second part.
Yep.
So can you put the slides on the screen and we can go through them one by one?
Okay. Slide number 1. We're not done yet. Optimal dosing pro-drug delivery of 6-diazo-5-oxo-L-noreleucine.
All right. So, So the DON is a substance that it has been discovered several decades ago, and it is been revisited periodically regarding as a potential treatment for cancer. And what I put here is the last review by an authority in the field, Barbara Slusher, regarding DON as a drug for solid cancers, and it addresses the optimal dosing and what actually happens to its body. And what I'd like to point out from that reference is that DON is a so-called promiscuous drug, because this is just some of the metabolic pathways that it inhibits. But the one which is very relevant that I have been discussing before, and the one that, of course, Professor Seifried is very interested about, is the pathway of glutaminolysis, which I have underlined with red. Now, as you can see from this figure, I'd like to draw your attention that DON is a substance that can inhibit glutaminase, specifically the isoform 1, and by doing that, it is supposedly able to deprive the tumor from utilizing externally added glutamine. However, if you see on the slide 2, this is something that I have assembled. I apologize for the overall details in the slide, but I'd like to have them as a reference for those who are interested later on to see the actual pathways. I have indicated with blue the particular enzyme which is inhibited by DON on the top right.
Right.
So the idea is that glutamine is found in the bloodstream. In humans, it is practically the amino acid which is mostly metabolized, meaning that and it's taken from one tissue to another. So there is a very high amount of glutamine in the bloodstream. Glutamine is up; it's taken up, and then within the cell, and this is actually a mitochondrion. Glutamine will break, let's say, into glutamate, and from that point on, there is a very important bifurcation. The one bifurcation is labeled in green, and the other part is labeled in red. So in this bifurcation point, one particular metabolite which is of utmost interest is alpha ketoglutarate, which I have indicated in this yellow box here and labeled as AKG. So this alpha ketoglutarate is the branching point, which is primarily coming from glutamate, which came from glutamine, and can give you something which is used for the tumor. In green is the fatty acids, or it can follow a so-called catabolic pathway, which is labeled in red. Now, DON is inhibiting an enzyme that breaks down glutamine to glutamate, but what you can appreciate from the figure is that there are other pathways that can lead to alpha ketoglutarate formation, the AKG formation. So DON is good because it will somehow diminish the provision of glutamate and alpha ketoglutarate prior to the bifurcation point, but it is not enough because you have other pathways that will lead into alpha ketoglutarate, and then it can follow the green or the red pathway. So this is what I meant, that the using of DON will not be sufficient to block the glutamate or glutamine catabolism in the tissue, because cancer cells have the capacity of getting alpha ketoglutarate from other intermediates as well. And cancers are really good in doing this. This is a very well, I wouldn't say known, but discussed phenomenon, which is called the metabolic rewiring. The metabolic rewiring is a phenomenon by which when you have pathways and you have a blockade in one pathway, then they're able to interact in some way in order to bypass the blockade. And tumors are excellent in doing this. So and this is a critical example, if you block glutamine catabolism by DON, the tumor cells will, for example, follow another pathway, like if you see down at the right, the GOT2 or MDH2, and they will be able to gain provision of alpha-ketoglutarate from other extramitochondrial or extracellular metabolites. So that's what I wanted to point out regarding DON.
Okay. Excellent. Should we move on to slide 3?
Yes. So the next slide refers to the other thing that I mentioned a little bit in the previous interview, which regards the carbohydrate consumption in cancers and how do tumors use glucose. So it is very central. It's a very central concept that tumor cells use carbohydrates as both energy, let's say, and building blocks, and especially glucose. And let me put this a little bit into a biochemical concept. Normally speaking, when we eat carbohydrates, and when we mean carbohydrates, we largely mean glucose. Of course, there are other type of molecules, complex and non-complex, like fructose and so on. But in the vast majority of cases, most of the carbohydrates that we consume will become glucose in the blood. And this glucose in the blood will be taken up through the cells by so-called glucose transporters, and then it will follow the most ancient biochemical pathway called glycolysis. Now, in glycolysis, in normal cells, you get a little bit of energy, you get a little bit of side intermediates which are used for biosynthetic pathways. Not too much, of course, it highly depends on what kind of cell we are talking about. And but the vast majority of the carbon molecules in glucose will become pyruvate, and that will follow pathway towards mitochondria and be burned, be used for oxidative phosphorylation. However, on the fourth slide, what actually happens is a modification of the glycolysis pathways in tumor cells, and I have also indicated here that this pathway can also be found in normal fetal stem cells, and it is true that tumor metabolism looks like a little bit like fetal metabolism or stem cell metabolism. Of course, in the latter two cases, it's coordinated in a good way, unlike in the tumors. And the glycolysis pathway in the tumors is extremely high. But the reason for that, it's not for generating energy, but it's for generating many metabolic intermediates which can be side passed to other pathways, like so-called the PPP, that's the pentose phosphate pathway, or the biosynthesis of other stuff, which are very much needed for rapidly dividing cells. Now, the tumors achieve this by two means, not just by activating the glucose uptake and the glycolytic pathway, but by also blocking their terminal enzymatic reaction. Because by blocking the terminal enzymatic reaction, which is indicated by an enzyme called PKM2, this is a very slow operating enzyme in tumors, they achieve that there is a very high metabolic traffic jam in the upstream metabolic pathway in glycolysis, which means that the metabolic intermediates are diverted into other intermediates required for biosynthesis. Now, this very high uptake of glucose in tumor cells has been known for decades, and it's actually used in diagnostic procedures of the PET scan.
Yep.
So in the PET scan, a subject will be given fluorodeoxyglucose which is able to be picked up by the PET scanning machine, and it would light up in places where there is very high uptake of glucose in the cell. Now here you will also see some other organs like the heart or the kidneys or the bladder, because the molecule will concentrate there as well, but it will also light up in places like around the neck, lymph nodes, that they exhibit tumor metastatic sites. So this increase of glycolysis and the need for glucose is central to the concept of the low carbohydrate diet, and in particular ketogenic diet, in the fight against cancer. Now, why this is good and why this is not enough? Well, it is good, obviously, because as it's the metabolic intermediates of glycolysis are terribly needed by the tumors to build new tumor cells and to metastasize. But why isn't it enough? It's because it is impossible to decrease blood glucose concentration to a level that you cannot favor even a little bit glucose uptake by the tumor cells. And to add up to that, the body expresses certain pathways, like the pathway of gluconeogenesis, which means that it can form glucose from non-carbohydrate molecules. For example, the proteins. Proteins consist of several amino acids. Some of them can be converted into glucose molecules very well, and this is a normal process called gluconeogenesis. And this is what prevents you from collapsing when you haven't eaten carbohydrates for many days. And so there will always be some level of glucose in the blood. Now, admittedly, it will be less, but it cannot be zero, and it cannot be less than the mechanisms found in the tumor, which are able to capture any glucose molecule found in the blood. So this diminished glucose consumption is important against cancer, but again, it's something by itself not sufficient. Now, what I have also included in the figure, that will be a little bit too much for a layperson, is that many people know, and this is true, that tumors form a very high amount of lactate. And this very high amount of lactate can only come from pyruvate. But then there is a conundrum here, that if we say that tumors express very high rate of glycolysis, but the terminal enzyme is inhibited, which means they have very low formation of pyruvate, then where does the lactate come from? Well, this is because tumors have more than 80 metabolic pathways that they use as rewiring in order to form pyruvate using metabolites other than glucose, because they need the lactic acid, not just the lactic acid for acidifying the tumor microenvironment, but they also need the regeneration of NAD, which is an oxidized form of a reducing equivalent that can be used to run glycolysis and many other things as well. So in brief, that should answer the question why carbohydrate consumption is relevant to cancer and how the tumors use glucose.
Yeah. Can I just ask, when I've seen many of Professor Seyfried's videos, gluconeogenesis, he said that if people exercise, then the cancer's not getting the glucose. The muscles are getting any glucose that's in the system. And the body and the brain, etc., are running off of ketone bodies.
Okay, so there is a very high competition for glucose between the exercising muscle, and the cancer. So it is true that when people go into ketogenic diet and at the same time they exercise, the brain will prefer the ketone bodies, and the muscle, especially if it is a kind of aerobic exercise, will use fatty acids and also take up the glucose, and that would diminish the amount of glucose that can be provided to the tumor. That is true. But my opinion is that you cannot exercise to the point that you will deprive the tumor from obtaining sufficient amount of glucose in order to form metabolic intermediates through the glycolysis pathway. And one, I think, finding to attest that this is the case is because low carbohydrate consumption by itself is not curative against cancer, no matter how much you exercise. It is not a way that can cure cancer. It can prolong, let's say, a disease-free interval, but it is not sufficient by itself.
Right.
The very
Yeah.
same-
No, I understand that, and Professor Seyfried does talk about the press pulse concept, which is his way of the metabolic therapy being the press pulse, which starves the cancer of what he says is both fuels that the cancer cells use. And obviously, from what you're saying, you're stating that it will still survive.
He is entirely correct in his thinking in that the tumor has to be deprived from its fuel, which is glucose and glutamine. That is, I'm 1000% concurrent with this. But what I'm telling you is that it's not possible to starve the tumor in a body by not eating carbohydrates. So you are able to diminish blood glucose concentration, but I can assure you that the glucose uptake mechanisms and the glucose utilizing mechanisms in the tumor will fight it to the point that it will be able to obtain glucose to an extent that will propagate the disease, albeit at lower levels. Yes, it's true.
Yeah.
But it won't be able to stop a tumor.
Right. Okay. I did share with you Dennis Stacey's video that I posted on my channel. It's just the half an hour version. The full version is linked in that video with the guy, Drew Porritt, was the guy that originally did the interview. And he did a press pulse protocol, and his whole protocol is on my website for you to take a look at. But he is now no evidence of disease, and he says he's N of 1, and he says it works.
Well, okay. So I looked at the video and I looked at the protocol. So as being a medical doctor myself, and I looked into this very rare tumor, and I mean I know where to look, not just whatever misinformation you can find on the internet. So the median survival rate from a sarcoma of the intima of the pulmonary artery is 17 months, and by median we see that's the middle. Okay, so
Yes.
that means it can be as little as 3 months, can be as high as many years, and the longest survival is 8 years. And I think the age of that video that I have seen, it was just a few months old, if I remember correctly. So this person is well within the expected life expectancy, let's say, of such a tumor. I mean, if the press pulse works and he is cured and many other people are cured, then I will be a very happy person. Of course, I will lose my job, but I will be extremely happy if it happens. So right now, I think it's in the wait and see. That's all. That's what I could.
Yeah, wait and see. There are many other people that I know of that are also going down that route. A lady by the name of Alison. She has a blog called Walking with Warburg, and she she was given months to live, stage 4 breast cancer that had metastasized to the bones. She was able to get DON, and she did the full press pulse protocol, also using drugs like Metformin to further reduce her blood sugar level. And when she did a 1 month protocol, so she was doing the whole press pulse thing. So she was pressing the glucose continually, and then was pulsing with DON and other substrates. And after the first month, her primary breast tumor totally disappeared. It was gone, and she is now still battling because obviously she's got metastasis to the bones. And I would be interested to for you to read the paper that's coming out of Siegfried's lab, with the first name on the paper being Thomas Durage.
Yes.
Have you read that paper?
No, I have not read that paper yet. I just want to tell that from what you said now, it's a very good combination to use DON and Metformin, because Metformin is something that will not just lower blood glucose level. That was the original marketing of Metformin, but Metformin is also a suspected complex 1 inhibitor. That is a polypeptide complex in the mitochondria, and there are many indications that even if there might not be oxidative phosphorylation in mitochondria, this particular complex may still be operating to a certain degree and continue and contribute to cancer cell survival. And if Metformin really inhibits this, that is actually good against cancer. So Metformin, I'm highly supportive of using Metformin with other drugs against solid cancers such as the breast cancer.
Yeah. So you're obviously fully on board with the metabolic theory of cancer as opposed to the genetic theory of cancer.
Yes. So I also wanted to mention, to explain my bias towards the metabolic theory of cancer, which is not just a bias towards the metabolic theory of cancer, but it's also an aversion from the genetic theory of cancer. And regarding my aversion for the genetic theory of cancer is because of the following two reasons. One of them is that the past few years, maybe decade, there has been an explosion regarding the cataloging of mutations in cancers.
Yeah.
And there billions of dollars have been spent. Tremendous amount of machinery and hands are in use for this. But what has been found are two important things. One is that many cancers, not all cancer, but many cancers, they do not express these so-called important mutations, which are best known as driver mutations. So these are cancers that don't express the mutations that people say are important for the cancer to develop. And the other thing is that these very same mutations that people find in cancers and they say they are important, they also find them in non-cancerous tissues. Yeah. And there are actually big profile studies by serious people that they say, well, we may have been a little bit overboard in reporting mutations and telling that these mutations are important for developing cancer. I'm not saying that there are no mutations which are important for cancer. There may be some, but in the vast majority of cases, you do not have driver mutations in cancers that are actually driving cancer. It's a misnomer, and it's unfortunately being propagated. And so this is my aversion from the genetic theory of cancer. The bias towards the metabolic theory of cancer There were some experiments approximately 50 or 60 years ago in which people were culturing post-mitotic cells. A post-mitotic cell is like the neuron or the cardiomyocyte. So when you are born with a neuron, you die with the same neuron. It does not divide. Okay. So they exposed post-mitotic rat cardiac myocytes to chronic hypoxia, but chronic hypoxia meaning like for 5 to 6 months. Now, these experiments are very hard to make because when you have a cell culture, in order to maintain it in sterile conditions for such a long time is a very big challenge. It gets infected. Everybody who has done cell cultures will consider what a huge feat is to maintain cell culture for 6 months. But what they have reported is that they started to see malignant transformation in post-mitotic cells if they were culturing them for 6 months under hypoxic conditions. So there was the hypoxia inducing a post-mitotic cell to somehow malignantly transform. So that tells me that that the metabolic part is not just important, but maybe casual as well. And in the past couple of decades, there has been many metabolized realized to act not as metabolized in biochemical pathway, but they have so-called epigenetic roles. An epigenetic role is something that it may act on a DNA or RNA, and it has a role other than its role in overall metabolism. Now, it is important to remember that when we talk about subjects like biochemistry or molecular biology or physiology, these are man-made concepts in order for people to better understand what is happening and how to teach that. That's why a medical student will have a concept, will have a class on biochemistry, will have a class on physiology, and so on. But these are all practically the same thing. So when people realize that metabolites, such as, for example, the alpha-ketoglutarate that I have shown in the figure before, has a epigenetic role in dictating the cell division, that practically means that metabolism has a say in the overall cell proliferation pathway. So that's why I think that the metabolic theory of cancer is much more important than previously thought, and that's my bias towards it.
Yeah. The professor says that the problems in the nucleus are downstream epiphenomena of the ROS, the random oxygen species. The damage to the nucleus is a downstream effect.
The reactive oxygen species. Okay, let me tell you something personal about the reactive oxygen species. I had my PhD on reactive oxygen species, and as soon as I finished my PhD, I ran away from them as far as possible because they are chaotropic agents. It's if you want to find a very high number of false positives and false negatives, just work on ROS.
Right.
So what I can tell you is that ROS has been blamed for causing cancer. The lack of ROS has also been blamed for causing cancer. So it's impossible to decipher what the heck is going on. So from my personal experience, I can tell you specific targets which are important being attacked by ROS and they cause cancer, and some that are not. So therefore, you cannot really put a general blame on ROS if they are good or not. When I entered research, which was 30-something years ago, the ROS was a bad thing. But now we know that ROS is a part of regular signaling. So you do need the ROS for correct functioning. And it may be that in the intricate pathways, alteration of pathways in cancer, it is the lack of the ROS that may contribute into the disease, not the appearance of ROS that may contribute to the disease. And why am I saying this? And I wish to stress this. People should not take antioxidants in order to treat their cancer, or in the fear of not getting cancer, they take antioxidants. That may be a wrong thing to do. So, for example, there was a very large study, the results of which have been published a few weeks ago, if I remember correctly, regarding the use of selenium and vitamin E, and it was found out that it worsens prostate cancer. So it's not really advised to take such antioxidants for prostate cancer. It's quite the opposite.
Right, yeah. And obviously, you've seen the nuclear transfer experiments that the Professor Seyfried talks about all the time.
Yes. Yeah, so this is an old story. There is one person that on top of my head which continues the nuclear transfer experiments. This is studies by Kai Pareto, if I remember correctly. And not just nuclear transfer, but mitochondrial transfer experiments in order to deduce the role of nuclei versus mitochondria in causing cancer in non-cancer cells and vice versa. I think these are pretty good experiments, but right now to transfer entire organelles into a cell is considered not very elegant. I'm not telling that it is stupid. It's actually good, but it's not elegant because we know the individual parts of mitochondria and individual parts of the nuclei. So it is too dull or too big, let's say, if you want to transfer an entire organelle into a cell, and because you are not able to make very concrete conclusions. Your conclusions are based on doing something that carries by itself very many confounding factors. So when you transfer, let's say, a nucleus, you transfer 10,000 things from within the nucleus. It's not just one thing that you are doing. It's not just one alteration.
Yeah.
Now with all these biochemical and molecular biology approaches, you can have a very, very small amount of alteration, and you can check what is the effect of this alteration in your, let's say, in your cell in culture.
Yeah. The professor does say that that it is a really difficult experiment, but it has been repeated. So
that's yes.
Well, Christos, I'm really glad that you've managed to get it off your chest. The questions that your peers were asking. I really would like to interview both yourself and the professor at the same time. It will all go over my head, but I'm sure it would be of very great value to the audience.
Yeah, I'm into that. I would like that too. It's really nice to talk with him. He really gets into it. So I love to see this. So.
Thank you for coming on again. And if there are any other further questions that people raise, then we can do a part 3. And in the meantime, I will contact Professor Seyfried and see if we can set up a meeting with the 3 of us.
Okay, that will sound good. Thank you very much.
Brilliant. Thank you. Cheers, Christos.
Bye.
Bye.